Friends, myself Jagdish Kara, lecturer in zoology, KB DAV College, Nirakarpur. Today I am going to discuss about production of human insulin by genetic engineering method. Insulin is a hormone. It is produced by the pancreas gland. The islets of Langerhans, a specific type of tissue present in the pancreas, produce insulin. The beta cells of the islets of Langerhans produce insulin. Insulin helps in regulating blood glucose level. When this regulation mechanism fails, diabetic condition results. Friends, nowadays number of diabetic patients are increasing. So, we require more amount of insulin for their treatment. Friends, for the first time Banting and Best isolating a protein from the isolates of Langerhans and injected it into diabetic dog for treatment. Since then, insulin has been used to help diabetics live out their lives. However, this insulin has its problem. For many years, insulin was extracted and purified from either porcine or bovine pancreas, that means from the pancreas of pigs and cattle. But obtaining insulin by this method has two difficulties. Though the animal insulin was chemically similar to human insulin, but there were some differences. These differences lead to antibody attack and inactivation as well as inflammation in many patients. Although there was the problem that this method of extracting insulin from animal organ made it difficult to obtain large amount of pure insulin. But nowadays it is possible to produce large amount of insulin as per our requirement to treat diabetic patients by using genetic engineering methods. Let us go to the parts of genetic engineering method. Let us first discuss the DNA and encoding of a protein. It is well known fact that DNA contains genes which encode all the different types of protein that an organism can produce. That means the DNA has the genetic code which is used for the synthesis of different types of protein in the living body. In 1955, Sanger for the first time sequenced insulin and determined that proteins are composed of amino acids. The amino acids are linked to each other by a peptide chain. The amino acids are linked by a peptide bond or known as peptide linkage. But it was not possible to produce large quantity of insulin till 1979. But now it is possible to produce large quantity of insulin by using recombinant DNA technology. Recombinant DNA or rDNA is a DNA which specially encodes a protein, is prepared as per need to produce specific proteins, antibiotics or vaccines. Let us discuss the steps in insulin production. The various steps of insulin production are: the first one is identification and isolation of insulin gene. That means human beings have insulin gene in their pancreatic cells, that means in the beta cells. Human insulin producing gene is identified and isolated from the beta cells of pancreas by using restriction enzymes. The restriction enzymes are used to cut the genomic DNA into fragments. Genomic DNA is cut by restriction enzymes which cuts DNA at specific sequence along the chain. These pieces are then analyzed and the DNA needed to make the protein is extracted and purified. Since insulin contains two polypeptide chains linked by disulfide bond, two pieces of DNA are extracted. That means insulin contains two polypeptide chains. The two chains are held together by disulfide linkage. So, two pieces of DNA are extracted to produce two polypeptide chains. These DNA strands are then placed into two different plasmids. That means two DNA segments are isolated and they are placed in two different plasmids. Friends, plasmid is used as a vector to carry the foreign or desired DNA. The second step is extraction of a plasmid DNA. A plasmid DNA is extracted from a bacterium and cut with a restriction enzyme, forming plasmid vector. The plasmids are then incubated with weakened strain of E. coli. That means plasmids are extracellular DNA which are isolated and cut by restriction enzymes. These are used as vector. Then these plasmids are incubated in the weakened strain of E. coli. The next step is production of a recombinant DNA. Human insulin producing gene is attached into the bacterial plasmid vector by using ligase to form the recombinant DNA of human insulin producing gene. That means in this step recombinant DNA is prepared. In this process insulin gene is attached into the plasmid vector by using ligase. The ligase is an enzyme which is used to join two DNA segments. Friends, these are the steps in preparation of recombinant DNA. Let us go to the picture. Here, this is the genomic DNA which is obtained from the beta cell of a pancreas. Then this DNA is fragmented by using restriction enzymes. These are the DNA fragments. Similarly, the plasmid is cut by restriction enzymes. Then the plasmid as well as the genomic DNA are linked by using ligase. Then the prepared recombinant plasmid is known as recombinant plasmids, or that is called as recombinant DNA. Let us go to the fourth step, introduction of recombinant DNA in the host bacterium. The recombinant DNA is then introduced into a bacterial cell to form the recombinant bacterium. That means when the recombinant DNA is introduced into the bacterium, then the bacterium becomes a recombinant bacteria. Since only one of the bacteria will take up the plasmid, a gene encoding an enzyme which breaks down a certain antibiotics is also included in the plasmid. That means antibiotics are used to incubate the plasmid. This process follows and it allows bacteria to grow along with the plasmid. The bacteria grow on the plate containing the antibiotics while the other bacteria die, because we require the genetically modified bacterium, not the other. So the modified one survive and the other tends to die. The next step is cloning of recombinant DNA and multiplication of a recombinant bacterium. The recombinant bacteria then allowed to multiply in a fermenter tank. That means the recombinant bacteria are cultured in a fermentation tank to produce large number of bacteria. This method of obtaining large number of bacteria by means of bacterial reproduction is known as cloning. These bacteria they are then allowed to grow and replicate, which allows the plasmid and the insulin gene to replicate millions of times. That means now the recombinant DNA is copied or cloned million of times. Friends, these are the methods in insulin production. As we have already discussed, this is the human pancreatic cell, then it is the obtaining of DNA fragment which encodes insulin, then here is the plasmid, it is cut, then a recombinant plasmid is formed, then the recombinant plasmid is introduced into the E. coli. E. coli is allowed to multiply in the fermenter, then the E. coli grows and secrete insulin which are stored in the cytoplasm of the bacteria. Then the bacteria are collected, their wall are lysed or destroyed, and purified insulin is collected. The next step is production of insulin. The bacteria produce insulin. We have already discussed it. Then the bacteria are given signal to produce protein and insulin identical to that of human can be produced. That means as we have produced the recombinant DNA, insulin along with other proteins are produced, but we need the insulin, so we collect it and purify it for our purpose. The next step is known as downstreaming process. In this process, insulin is extracted, purified and bottled, and it is then ready to be injected into the diabetic patients. That means now we are able to get large quantity of insulin as per our need to treat our diabetic patients and to save their life. This insulin produced in the bacteria is named as humulin. It is used to treat people with type 1 diabetes without the worry of allergic reaction. Thanks.